for about 40 years, we worked independently in very different fields. And then, probably about 15 years ago, we realized that we had an interest in common, which was in the physical laws that governed the natural and the man-made worlds, particularly this issue of inequality in both social and natural systems that seems to be pervasive. And Les had been doing some research on this, and he'll explain that in a moment. And Les was very interested to move this research into the area of the biological sciences, because apart from anything else, the biological sciences have often been thought to be so complicated and so specialized that they were somehow exempt from many of the laws that would otherwise govern natural systems. But I'll let Les take it from here.
Yeah, thanks, Greg. All this started for me in about 2008 when I was studying the failures in software control systems. And that eventually caused me to have to break down computer programs into their component pieces. This led to insights about how systems fail, but the results that I was getting showed that there were commonalities between systems which we didn't really believe should have commonalities. So, for example, if you analyze computer software written in different programming languages for different purposes, they show the same behavior. Greg and I have come to call this emergent behavior. I got so far, a colleague at my university suggested I should look at biology. I know nothing about biology, slightly more now, but not much. And I contacted Greg. It was nice to catch up anyway, and chatted to him about it. And that was the start of certainly the most productive collaborative research of my life. So ever since about 2013, we have been merging Greg's skills in biology, biological sciences, biochemistry, and just general all-round knowledge with my skills in computing, mathematics, and information theory, and found that there is, as is often the case in science, the areas between disciplines are often the most productive. And it's been very productive for us. The only problem is, which we'll get to, is that journals find it hard to review interdisciplinary papers. So it's sometimes difficult going, trying to get results published. But we eventually got there thanks to the auspices of excellent journals like your own. And we're still at it after 13, 14 years, and often feel that we've hardly scratched the surface. So it's really a sort of a marriage of two very different disciplines, which just happens to work really well. And it's underpinned by this very long friendship we've had.
Yes. Again, I'll just back up a little bit and say that I think the two most rewarding things about a career in scientific research are, first, the opportunity to see things that nobody else has seen before, and second, an opportunity to see how beautifully, logically, pieces of a puzzle with which you're challenging yourself can fall into place. And one of the ways that Les and I approach our research is to try different approaches which are independent, and if the results fit nicely together into a logical story, then that is two pieces of a jigsaw puzzle that again help us to feel that we've got a handle on the correct answer. And in this case, a typical way that we would first approach the question of these patterns in the transcriptome is by analyzing the frequency of short words of bases in the DNA, the so-called KMers. We would look at their rank order distribution, and we would also look at a phenomenon called inverse symmetry in the DNA, which actually was discovered well over 60 years ago by Erwin Chargaff, and was, I think, only recently explained as a result of the interaction between the conservation of Hartley-Shannon information and what's the familiar Watson-Crick base pairing between the two helices of the two strands of the DNA helix. So in this particular experiment, we were looking not at the KMers. We were looking at specifically how the reading frame, the manner in which you could translate a single coding sequence differed whether you looked at it in its natural, appropriate, correct reading frame or in one of the incorrect reading frames. And the control was non-coding RNA, ribosomal RNA, one of the many other species of RNA that occur in the cell. And when you compared two reading frames in that sequence, interestingly enough, you find that the frequency of the codons is essentially identical, whichever frame you're looking at. But when you come to the coding sequence, the result is very different. The coding and a non-coding frame show very different frequencies of all the codons. And in particular, the stop codons are very distinct. They're about 10-fold difference in their frequency in reading frame and out of reading frame. And this makes complete sense because the stop codons are probably the most powerful signals in the code, in the genetic code, in that they just produce an absolute stop to the translation. So it was very satisfying to find that result. It's what it boils down to.
Thank you. Basically, I studied how computer systems fail for about 25 years. The picture is not very good for science, in that I believe that most scientific software is corrupted by unquantifiable errors, and I have evidence for this. So this is obviously an interesting area to pursue, but I found myself starting looking at systems as a whole and gradually going down into them to look at the component parts of the computer programs themselves, in a very similar manner to an author with a book. You can break a book down into chapters. You can break a chapter down into paragraphs. You can break a paragraph down into sentences. You can break a sentence down into words, and so on. So as you look deeper and deeper, I was looking to find patterns in the software which might correlate with failure. The ultimate goal of which is to predict whether a computer program will fail or not. I succeeded in analyzing the software, and I failed in trying to produce any sensible correlation. However, one of the things which emerged, and this relates to a long-standing issue in computer software, is that the lengths of the components in any software package always obeyed the same relation. Now, if you've worked in computer science, you would know that this is a very odd result, because we spent many, many years coming up with how to structure your software and all the rest of it, which would change the distribution of lengths of components. But it doesn't. If you look in the data, whatever the language, whoever wrote it, and whatever it does, they're all the same. So there's obviously, to me as a mathematician, the whole thing smelt of a conservation principle. And I used to study statistical physics a lot. So I just felt there was something going on underneath. It took me about 4 months to prove what we call the asymptotic version of this, which did indeed predict that the length of software components is independent of the meaning of the tokens. It only depends on the fact that you can distinguish between the pieces. They're called tokens in computing. And this was a really fascinating result. I finally published this in 2014, I think. But there are other odd features of this distribution, which are also generic. They apply across many different systems. And at this point, an idle conversation with a colleague of mine in my last university said you should look at biology. And I know nothing about biology. I know a little more now, thanks to Greg, but not that much. I approached Greg, and he, being Greg, he got it straight away, and we started delving deeper, and we found that the length. One of the first results we found, which is predicted by this theory, conservation of Hartley-Shannon information, was that the lengths of proteins is also independent of the meaning of their constituents, which are amino acids. This is a big result in that the diversity of possibilities in a protein of, say, 400 amino acids is so enormous that you wonder why on earth would life need a longer protein. And yet, even then, we were finding proteins of more than 25,000 amino acids, way, way longer than is necessary biologically. And we made a prediction in 2015 that within 5 years, we would see proteins of more than 40,000 amino acids, because the distribution is effectively scale independent. And sure enough, 5 years later, the first protein longer than 40,000 amino acids was discovered. So it's clear without any shadow of a doubt that one of the fundamental properties of life, the length of the proteins which drive it, has nothing to do with natural selection. This is a very big statement. Just as in computer science, it's a very big statement to a programmer saying that they have no influence over the lengths of components that they produce. Just as a side issue, maybe you may know about this, but there's a famous law called Zipf's law, and it was discovered nearly 100 years ago by the American philologist George Zipf, and he noticed that the frequency of words in books always obeyed the same shape. And it didn't matter whether the book was written, what language was written in, who the authors were, what the subject was. There is an unseen hand guiding the distribution of the words as we write. And this is a very spooky thing indeed, if you didn't understand what was going on. One of the things we managed to prove on the way was why this occurs. We managed to prove Zipf's law, why this occurred in books. So now we have several different discrete systems, computing science and computer programs, the written word, and the enormous domain of the genome, the proteome, all obeying the same emergent principles. And from then on, we've just carried on mostly, even though we've demonstrated these principles in other areas, for example, finance, the distribution of wealth obeys exactly the same principle. Although it occurs in many areas, we've been so busy with the enormous domain of life that in the last 10 years, we've almost entirely reserved ourselves to that. And finally managing to complete another couple of proofs on the way. So looking back, the merging of the two disciplines, Greg's and my own, seems so natural as to be obvious, but it wasn't obvious at the time. Science never is. When you look forward, you're always looking into a fog and you just explore away and suddenly you find something and you wonder why. And then you just keep going like that. And then we finish up where we are today.
Perhaps just jump in here and take up the story from where Les left off. This pattern that Les mentioned that was originally discovered by George Ziff, we knew at the start of this particular project that this pattern existed in the genome as a whole. And of course, the human genome, the coding region is only a few percent, perhaps 2%. So the question was, does this pattern, the one that was discovered by George Ziff, does this exist also in the coding region? Because the coding region has many completely different functions from the rest of the genome, in particular, the necessity to code functional proteins. And one of the key discoveries of our paper was that this pattern is preserved in the coding region, even though, as we mentioned earlier on, you get this disruption in the ratios of the various codons and the stop codon. So this conservation principle is extremely powerful in guiding biological systems. What we also discovered, the second point I would say in this paper, is that the power law distribution, this Zipfian distribution, in DNA results in inverse symmetry as a purely secondary property. And that's widespread throughout the genome if you just look at the whole, say, the human genome. But when you look at the coding regions, as we did, the inverse symmetry disappears. And so the necessity to code functional proteins appears to have degraded a secondary property of the DNA that is involved in coding, but not this primary property which is guided by this conservation principle. Yes, if I could just go first, without wanting to sound too self-satisfied, I think that the approach we've had has been extremely successful in analyzing systems and explaining how biological systems, indeed all discrete systems, as Les mentioned, and a discrete system is one that's made of countable pieces. We've been very successful in making predictions about the distribution of properties that should be found in discrete systems. And we've been pretty successful also at identifying where particular pressures specific to the system cause perturbations in the equilibrium that's predicted by COSY. But what we would really like to do is to be able to predict where these discrete systems are going and why they're going in particular directions. We can explain why they've reached equilibrium and where particular perturbations in that equilibrium have been caused. But where we can't readily predict, we can't predict at all on the basis of the mathematics is where they're going. We can't predict the future course of biological evolution, unfortunately. I'm sure Les has some thoughts on this.
I was just looking at the notes I made for this interview, and I just noted at the end of this question, too many for a lifetime. Every time we do something, it uncovers two more interesting things. So the project, it's one of the reasons it's been going for so long. And I think we have, I was counting up on my systems about a month ago. And so far, in the last 15 years, this project has produced 35,000 figures, most of which we've never used. So it's been really a very, as Greg says, a very fertile area. And given the generality of discrete systems, there seems so many other things that are feasible to do. Real insights. For example, as we speak, Artemis is on its way back from the moon. It might interest you to know that the widths of craters on the moon follow the same distribution as the genome. There are two very different discrete systems.
If I could, again, wax perhaps on the sociology of science. Many fields of science are specialized in the journals in which people publish, and certain schools of thought come to dominate the way that data are interpreted and what is considered publishable in those fields. And Les and I have encountered a great deal of this in the course of this collaborative research and the publications we've undertaken. And it can be very frustrating when you have what we think is a new and rather challenging way of looking at biological systems. As Les mentioned earlier on, it does question the role of natural selection in generating these patterns of nature. They seem to arise mathematically, regardless of cause. And so we were very happy that Frontiers in Bioscience-Landmark reviewed very carefully, very objectively, the science we submitted on its own terms, rather than trying to fit into a framework of a preconception of what it should look like, what the paper should look like, and what the results should look like, how they should be interpreted. The review was good, objective, and to us to be directed towards the question of, can we make this paper the best it can be for publication, which is I think the ideal way that paper should be read. As and I have had a lot of experience of editing journals over the years, and this was one of the best experiences that we have had as authors submitting manuscripts for review.